Influenza. Influenza is the respiratory disease caused by influenza virus, which belongs to Orthomyxovida. This virus has been responsible for millions of deaths worldwide and epidemics.
The types of influenza viruses. There are 3 immunologic types of influenza viruses, designated A, B, and C. Antigenic changes occur within the type of influenza viruses and to a lesser degree. In the type B group, type C viruses are antigenically stable. Influenza A strains are also known for aquatic birds, chickens, ducks, pigs, horses, and seals. Some of the strains isolated from animals are antigenically similar to strains circulating in the human population. Structure and composition of influenza virus. It is spherical and about 100 nanometers in diameter, 81 to nm. The genome is single-stranded. Negative sense RNA in influenza and B viruses exist as 8 separate segments. Influenza C viruses bear 7 segments, however, lacking a neuraminidase gene.
The virus contains 9 different structural proteins. The nucleoprotein NP associates with the viral RNA to form a ribonucleoprotein RNP structure, 9 nm in diameter, that assumes a helical configuration and forms the viral nucleoside. 3 large proteins PB1, PB2, and PAR are bound to the viral RNP and are responsible for RNA transcription and replication. The matrix M1 protein, which forms a shell underneath the viral lipid envelope.
The virus-encoded glycoproteins, hemagglutinin H and neuraminidase Na, are found in envelope and serve as spikes about 10 nanometers long. These 2 surface glycoproteins determine antigenic variation of influenza viruses. The M to ion channel protein and the N ester protein are also present in the envelope.
Because of the segmented genome, when a cell is co-infected by 2 different influenza viruses, mixtures of parental gene segments may be assembled into progeny virions. This phenomenon, called genetic reassortment, may result in sudden changes in viral surface antigens. This feature of influenza and poses significant problems for vaccine development.
Resistance and sensitivity. The influenza viruses may be stored at 0 to 4C for weeks. Lipid solvents, protein denaturants, formaldehyde, and irradiation destroy infectivity. Both infectivity and hemagglutination are more resistant to inactivation at alkaline pH than at acid pH.
Classification and nomenclature of influenza virus. Genus Influenza virus A contains human and animal strains of influenza type A. Influenza virus B contains human strains of type B. And influenza virus C contains influenza type C viruses of humans and swine. Antigenic differences in 2 of the internal structural proteins, the nucleocapsid MP and matrix M proteins, are used to divide influenza viruses into types A, B, and C. Antigenic variations in hemagglutinin and neuraminidase are used to subtype type of viruses. The standard nomenclature system for influenza virus includes type, host of origin, geographic origin, strain number, and year of isolation. Antigenic descriptions of the H and the Na are given in parentheses for type A. The host of origin is not indicated for human isolates, such as a Hong Kong 368H3 and 2, but it is indicated for others, such as a swine, Iowa 1530th H1 and 1. So far, 18 subtypes of hemagglutinin, H1, H18, and 11 subtypes of neuraminidase, and 1 and 1,1, in many different combinations, have been isolated from humans and animals. Structure and function of hemagglutinin. The primary sequence of hemagglutinin H contains 566 amino acids. The H protein is cleaved into 2 subunits, H1 and H2, that remain tightly associated by a disulfide bridge. A hydrophobic stretch near the carboxyl terminal of H anchors the H molecule in the membrane, with a short hydrophilic tail extending into the cytoplasm. Oligosaccharide residues are found at several sites. The hemagglutinin H molecule is folded into a complex structure. Each linked H1 and H2 dimer forms an elongated stalk capped by a large globule. The base of the stalk anchors it in the membrane. 5 antigenic sites on the H molecule exhibit extensive mutations. The hospike on the virus particle is a trimer composed of 3 intertwined HA1 and HA2 dimers. The cleavage of HA1 and HA2 is necessary for the virus particle to be infectious and is mediated by cellular proteases. Influenza viruses normally remain confined to the respiratory tract because the H cleavage protease enzymes are expressed only at those sites. The amino terminal of H2, generated by the cleavage, is necessary for the viral envelope fusion with cell membrane. Low pH triggers a conformational change that activates the fusion activity. Function of the hemagglutinin hospikes is to attach the virus to the particular creators on the host epithelial cells. Structure and function of the neuraminidase. It is a tetramer of 4 identical monomers. There is a catalytic site for neuraminidase on the top of each head of the monomer. It functions at the end of the viral replication cycle. It is a sialidase enzyme that removes sialic acid from glycoconjugates. It facilitates release of virus particles from infected cell surfaces and helps prevent self-aggregation of virions by removing sialic acid residues from viral glycoproteins. It is possible that neuraminidase now helps the virus negotiate through the mucin layer in the respiratory tract to reach the target epithelial cells. Antigenic drift and antigenic shift. The 2 surface antigens of influenza undergo antigenic variation independent of each other. Minor antigenic changes are termed antigenic drift. Major antigenic changes in hemagglutinin H or neuraminidase NA, called antigenic shift, result in the appearance of a new subtype. Antigenic shift is most likely to result in an epidemic. Antigenic drift is caused by the point mutations in the gene, resulting in amino acid changes in the protein. Sequence changes can alter antigenic sites on the molecule such that a virion can escape recognition by the host immune system. Antigenic shift reflects drastic changes in the sequence of a viral surface protein caused by genetic reassortment among human, swine, and avian influenza viruses. Influenza B and C viruses do not exhibit antigenic shift because few related viruses exist in animals. Replication or life cycle of influenza virus. After receptor-mediated endocytosis, the low pH within the endosome is required for virus and membrane fusion that releases viral MPS into the cytosol. Acid pH causes a conformational change in the host structure, so that the viral ribonucleoprime complexes are released into the cytoplasm and transported to the nucleus, where replication and transcription take place. 1 messenger NAs are exported to the cytoplasm for translation. 2 early viral proteins required for replication and transcription, including nucleoprotein MP and a polymerase protein PB1, are transported back to the nucleus. RNA polymerase activity of the PB1 protein synthesizes positive single-stranded RNA-SS RNA from genomic negative single-stranded RNA SS RNA molecules. 3 these plus SS RNA templates are copied by the RNA polymerase activity of the PB1 protein. For some of these new genome segments serve as templates for the synthesis of more viral mRNA. Later in the infection, they become progeny genomes. Viral mRNA molecules transcribed from some genome segments encode structural proteins such as hemagglutinin H and neuraminidase NA. These messages are translated by endoplasmic reticulum-associated ribosomes and delivered to the cell membrane. 5. Viral genome segments are packaged as progeny virions barred from the host cell. Pathogenesis of influenza infection. The virus transmits from person to person by airborne droplets or by contact with contaminated hands or fomites. A few of respiratory epithelial cells may be infected if deposited. Virus particles avoid removal by the Korff reflex and escape neutralization by pre-existing specific immunoglobulin IgA antibodies or inactivation by non-specific inhibitors in the mucus secretions. Progeny virions are soon produced and spread to adjacent cells, where the replicative cycle is repeated. Viral neuraminidase lowers the viscosity of the mucus film in the respiratory tract, laying bare the cellular surface receptors and promoting the spread of virus-containing fluid to lower portions of the tract. Within a short time, many cells in the respiratory tract are infected and eventually killed. The incubation period of illness varies from 1 day to 4 days, depending on the viral dose and the immune status of the host. Viral shedding starts the day preceding onset of symptoms. The infectious virus is very rarely recovered from blood. Influenza infections cause cellular destruction of the superficial mucosa of the respiratory tract, but do not affect the basal layer of epithelium. Complete reparation of cellular damage takes up to 1 month. Viral damage to the respiratory tract epithelium lowers its resistance to secondary bacterial pathogens, especially Staphylococcus, Streptococcus, and Haemophilus influenzae. Edema and mononuclear infiltrations in response to cytokine release and cell death caused by viral replication probably account for local symptoms. The fever and systemic symptoms associated with influenza reflect the action of cytokines. Clinical findings and symptomatology. Influenza targets mainly the upper respiratory tract. 1. Classic or uncomplicated influenza. Symptoms of classic influenza usually appear abruptly and include chills, headache, and dry cough followed by high fever, generalized muscular aches, malaise, and anorexia. The fever usually lasts 3 to 5 days. Respiratory symptoms typically last another 3 to 4 days. Mild or asymptomatic infections may occur. Coryza and cough may persist for several weeks. Children may have high fever and a higher incidence of vomiting. Influenza viruses are an important cause of severe croup in children of less than 1 year. Finally, otitis media may develop. 2. Pneumonia. This serious complication usually occurs only in old and debilitated individuals, especially those with underlying chronic disease. The deaths in influenza is caused by pneumonia and cardiopulmonary diseases. Influenza infection enhances susceptibility of patients to bacterial superinfection caused by Staphylococcus aureus, Streptococcus pneumonia, and H. influenzae. Combined viral bacterial pneumonia is more common than primary influenza pneumonia. Some S. aureus strains secrete a protease able to cleave the influenza hemagglutinin H, thereby producing more infectious viruses in the lungs. 3. Reye syndrome. Is an acute encephalopathy of children and adolescents, usually between 2 and 16 years of age. It is a recognized rare complication of influenza B, influenza, and herpes virus varicella zoster infections. Laboratory diagnosis of influenza. The diagnosis depends upon identification of viral antigens or viral nucleic acid in nasopharyngeal swabs and nasal aspirate or lavage fluid, and are frequently used for laboratory testing. Rapid detection of influenza NA-IN is carried out by using reverse transcription polymerase chain reaction RT-PCR. Antibodies against viral proteins HA, Na, MP, and matrix are detected by high hemagglutination inhibition and enzyme-linked immunosurban assay ELISA.